Hello everybody, this is Moritz Zaiss speaking, and I'm presenting the paper today for the MRM highlights. Downfield-NOE-suppressed amide-CEST-MRI at 7 Tesla provides a unique contrast in human glioblastoma. So this paper is about a CEST MRI, so I want to first give a short introduction to CEST MRI. CEST stands for Chemical Exchange Saturation Transfer, and in addition to the large water proton pool depicted here, we also have a small proton pool, which is here, for example, the amide proton pool of amide protons in the backbone of proteins. They have a much smaller signal compared to the large water signal. But what we can do in CEST imaging is we can saturate these protons selectively, and then this, by the chemical exchange reaction, this saturated state is transferred to the large water pool. So this state, this saturation accumulates in the water pool, and the longer we saturate, the more saturation of the water pool we get indirectly. And the difference between the unsaturated and the saturated image gives us the CEST effect, or a measure of the size of this chemical exchange process and the size of this proton pool, the smaller proton pool. This animation was created by Nirbhai Yadav from Johns Hopkins University. So what we actually do in CEST MRI is we take a fast MRI of the water signal, but before we have a RF saturation at different frequencies. So this is what we call a saturation. And if we do this for a lot of different frequencies, we get the so-called C spectrum. So this is the signal after saturation at a certain frequency offset or short offset. So if we are far off resonant, like here, we get almost a full water signal. So this is normalized to 1 here. If we hit the water directly, of course, we have no signal anymore. So this is the 0. But if we now hit these mobile protons, they exchange and we get this relatively small CEST dip. So a common analysis method is this MTR symmetry, which is just taking the right side minus the left side. Or there are better methods to use a baseline. We use the Lorentzian difference in this paper, which I will describe later, but in the end it's about isolating of the CEST effect, which is this small peak here. So in the end, it's, as I said, a difference technique, so between 2 images and saturated and the unsaturated reference image, and then we get the CEST contrast. The important thing is that this indirect imaging method has an enhancement factor called the proton transfer enhancement, and this is approximately the exchange rate of this process times T1. So if the exchange rate is 100 Hertz and the T1 is 1 second, then we have a 100 times higher signal compared to spectroscopy. And this is what makes CEST so interesting. So if we look in Vivo, we see that there's not only a single peak, but there are many peaks appearing. So this is a C spectrum acquired at 9.4 Tesla of red brain, and we see that there is an amide proton peak at 3.5 ppm. So this is what we're also focusing today. But there's also other peaks like amine or guanidinium protons around 2 ppm. There are OH groups, for example, of glucose or GAG or lactate closer to water. And they are all governed by their exchange rate. And this exchange rate depends on the pH, the temperature, and also the buffer situation. However, on the right side, there are also effects. These are so-called nuclear Overhauser effect, especially exchange-related nuclear Overhauser effect. So this is a 2-step process. So these are dipolar coupled spins, like immobile spins, but via dipolar coupling, they transfer the saturation to nearby, or like by spin diffusion over the whole molecule. This saturation is transferred to exchanging protons, and they transfer it to water. So for nuclear Overhauser effect, important value is this sigma, the cross relaxation, and this depends on the distance of the protons, the correlation time, and also B0. And it was also shown that this depends on the protein structure. So if the protein changes or aggregates, this can also be seen in this peak. So the CEST effect of interest in this paper was amide proton transfer, which was already published some years ago. So in 2003 there was this big article of Jinwan Zhu and Peter van Zyl in Nature Medicine, where they showed that you can use this amide proton transfer to detect cancer. And you see here already a correlation with the gadolinium, but we will see later that this can be refined. At the same time, the pH dependence can also be used in stroke imaging. So what you see here is that with lower pH, the APT quenches, and you get a signal loss in the infarcted region. So the big issue about this so-called amide proton transfer is that, especially if you do it at low fields, as I showed you, there are many different effects overlapping, and at low fields these peaks get very broad, and you can just do this asymmetry analysis and see just a plot. This is a plot of the asymmetry, and you just see that this is a mixed signal of different peaks. So if we go to 7 Tesla, what we can do is we can isolate these different peaks by a Lorentzian fitting, and this is also what we did in this paper. So what you can do is you can fit the waterline or a superposition of waterline, a semi-solid MT, then a NOE and amide peak and also an amine peak, and then you can use this amplitudes of the Lorentzians to create your image contrast. Yeah, and so this is what we tried also in this paper, to really isolate these amides, the amide proton transfer, and not just using the asymmetry. However, you can do this, and we were able to do this, but still there's some problems of the indirect measurement, because the water pool has relaxation parameters. Yeah? We know that the water pool also has different properties depending on the tissue, which is governed by T1 or T2 or also semi-solid magnetization transfer. And if you look into CEST simulations, you can find out that if you, for example, change just the water T1, so here we have a C-spectra of a long T1, long T2, and here a C-spectra of a shorter T2, and if you also add in the simulation MT, we have the CEST effect is artificially diluted. Yeah, so it's much smaller. We just see this here in Vivo, which is just the tip of the iceberg. Yeah, and actually CEST effect would be much bigger. So we have to take that, keep that in mind, that this CEST effect is affected. And yes, this is a so-called spillover dilution, and here it's like 70% of the effect is actually spillover dilution, which is governed by T2 and MT. And so we're really underestimating this iceberg, and this we know from history can have catastrophic consequences. So this can also be shown in a phantom experiment. So here we actually used the same creatine, same amount of creatine, same pH, same temperature. We just did a different T1. So here we added agar, we added some more agar, here we added gadolinium and more gadolinium, so to have the relaxation time down to 0.3 seconds, the T1 relaxation time. And the CEST image is very colorful, which, and if it would be a CEST effect, which correlates to the concentration, it should be a homogeneous image. And this can be explained by some theory. I don't want to go in detail here, so this is a complicated slide, but in the end you can use the spin lock theory, so CEST can be understood as a R1 decay, actually, a relaxation process. And what is important is that the C-spectrum, so this spectrum can be described by this formula. And the interesting thing is that the exchange-dependent relaxation, so this R exchange term, this induces the CEST effect, but it appears here in the denominator. Yeah? So if we look into this asymmetry evaluation, I also want to tell that this R exchange is also why we call this AREX, this signal. So we try to isolate this signal and call this the apparent R exchange signal. So if you look into this asymmetry and use this difference, then we see that this term in the denominator is actually responsible for this dilution. So it's in the denominator, and also the R effective and RMT, so this is T1 and T2 relaxation, they are all in the denominator. So this actually dilutes the signal. But what we can do is a simple solution. We just take the inverse difference. So this is what we call the MTR-RX change. And this is just RX change, this important parameter, divided by R1 of the water. And now we get back the intensity of the agar phantoms. Yeah? So T2 is already eliminated. And if we further multiply the signal by R1, we get back RX change and get the relaxation compensated CEST signal. And now it's not a colorful image anymore, now it's really a quantitative CEST image. And this is what we want to apply in tumors, and this was what we did in a previous study. We applied this in brain tumors, so we have our two-dimensional CEST sequence consisting of like a 4 seconds saturation and a fast centrically ordered readout. We all had to face B0 inhomogeneities, which were just measured by the B1 map, B0 map, and then corrected. We also had B1 inhomogeneity problems, which we solved by measuring multiple B1 and then interpolating that, so we could also get to this solved. And as I said before, we could isolate the effects by this multi-loop. So we applied this CEST contrast to 11 glioblastoma patients, and here you see one first patient where you see the conventional MR contrast in the top row, so gadolinium T1, ADC, T2 flare, and T2. And in the lower row you see MTR asymmetry, which already shows the tumor area. And we see the relaxation compensated amide-CEST at 3.5 ppm, so AREX, and we see also the AREX of NOE at minus 3.5 ppm. And we also see the T1 map. So what you can see here is that, we see something in MTR asymmetry, and in we also see something in the AREX 3.5 ppm, but they are very different contrasts. And if you see that the tumor area is actually hypo-intense in this map, but you also see that it's not so really the gadolinium contrast enhancement, which you see in figure A, is not correlating very much with AREX 3.5. At least it's not significant compared to contralateral white matter. So if you look at these two images, and we saw this in all the patients, that after correction of T1 and after we doing everything correctly, we don't see a contrast anymore. Yeah, so here you see the statistics for all the different patients on the below the figure. So it's not significant difference between contralateral white matter. Yeah, this is the contrast enhanced region. At the same time, the nuclear Overhauser contrast is very much decreased in the tumors, so you really see that this correlates with T1, but also all around the tumor area or the gadolinium enhancement, you see a lower NOE effect. So we were wondering, because this speaks now that this amide-CEST effect is not very significant, so but it still looks a bit weird, because it goes like first if you move towards the tumor, towards the gadolinium, it first goes down in the surrounding area and then it increases again. So this was a little bit weird. This looks a little bit like the AREX signal at 3.5 ppm behaves a little bit like the NOE signal at minus 3.5 ppm. And this was the beginning of this study I want to actually present, which is about this downfield-NOE compartment. So there was a paper or an abstract of Tao Jin at ISMM 2013, where they added D2O to BSA proteins, and they found out that there's an aromatic rNOE contribution also at the positive side of the C spectrum. So it's not just an aliphatic RNA, but also an aromatic, or we call it just a downfield NOE. So the aliphatic is the upfield NOE, and this is a downfield NOE. So they stated it first and also showed it in mice with very low pH regions, that there still are signals of the NOE. And we did the same in BSA. We went to different pH. So this is like VEX spectroscopy here, and we looked for. So the VEX spectroscopy just shows very slow exchanging processes, which are NOEs. NOEs are very slowly exchanging. And what we saw here is that in addition to the upfield NOE, we also see downfield signals, and we changed the pH in this BSA solution. And what we see is that the pH dependence is very much constant for both the upfield NOE and also this downfield contribution. So this is the first hint that there is a downfield NOE, or second hint after tau gene. So there's a second NOE signal downfield. We also have insights from folding experiments. So this is a paper in NMR and biomedicine, and what we did here is we unfolded BSA, and what you see is again that at the top image, you see that upfield NOE decreases with protein unfolding. So yeah, the more unfolding you have, the more this signal decreases. And on the left side, you see that also around 3.5 ppm, also the baseline decreases. Yeah, we see here exchanging peaks at 2.7 ppm. They are actually increasing, but the overall baseline, at 2 ppm, there's some aromatic resonances, but also up to 4 or 5 ppm, the whole baseline seems to decrease. So this is also an NOE-like behavior in this ppm range, so around amides. A further hint is given us from an experiment where we used pig white brain matter homogenate. So we took pig brain and homogenized it using a tissue grinder, that we have access to the intracellular compound. By this we have access to change the pH, so we could titrate the pH to different levels in this sample. And by that we also saw that the amide peak here at 3.5 ppm changes with pH, yeah, but we saw there's a constant baseline. So if we go to very low pH, we still see there are some signals left. And with this ratio, so we could estimate from the pH exponential fit, we can estimate this baseline and get an idea how big is the NOE, this downfield NOE. And we found out it's about 0.2 times the upfield NOE. Yeah? And we said, okay, we try to isolate this pH-dependent amide signal. Yeah? So this pH-dependent signal should be from mobile amides, but the rest should be a NOE compound. So let's isolate it in the following way. We define the downfield NOE-suppressed AREX, so the downfield NOE-suppressed amide. So this should be the pure amide. We define it as the AREX at 3.5 minus a fraction, and this is like 0.2, of the AREX at minus 3.5. So we remove the downfield NOE by using the upfield NOE. Yeah? That's the very first approach, but that's what we tried because we saw, okay, that's a similar origin, similar behavior, and both not so much pH dependent. So this is the actual measure we did in this study, and we could directly transfer it to a brain tumor. So here is a Roy evaluation, and here you see again the AREX signal in tumor and in normal white matter. And what you see, they are overlapping, and there's not a significant difference, but we see that there's some peak in there, and we see that the NOE on the other side changes a lot. So now we apply this calculus here and have the downfield NOE suppressed amide-CEST, and then we see actually in the tumoroid, we see this peak really coming out from the background, yeah, and the background is really lower. Yeah, so with this, we wanted to evaluate all the data, but first I want to show you that we also checked for other influences. So we did some control experiments. So first of all, we looked into if we do the tissue pieces. So for this, from this brain, pig brain example, if we do pieces of the brain or homogenate, does this change? Because it can change the mobility, but it actually did not change the CEST effects too much. Yeah? Then we also looked for different field strengths. So on the right side you see a plot of actually BSA, so just a protein solution at different field strength. And you see the effects are different. Yeah? That's mainly because the labeling gets more effective if the field strength is smaller, so you have a broader labeling, a broader pulse actually. But what you can see is that the ratio right side and left side is approximately the same. So our idea of using this ratio is still valid also for different field strengths. And in the last control experiment is just looking if this tissue homogenization changes a lot about the signal, but yeah, we saw that this is in time. So if we just wait some minutes and some hours, this is relatively stable. So there's not a big lysis process or protein denaturation processes going on. Okay, so we were quite confident that we're on the right track. This is not the final method, but it's a first step to get the real amide-CEST. And what we then saw in tumors, so we used these two contrasts, and now we calculate this new downfield-NOE-suppressed AREX, downfield-NOE-suppressed amide-CEST. We get a very interesting contrast, which was very similar to the gadolinium uptake in this patient, and if you compare it to the MTR symmetry, we think we get a much more interesting image here. And we saw this really in 10 of 10, so this is the number 11, but in the other 10 patients we also saw very similar structures in all the tumor patients. So here you see gadolinium on the left and dnsAREX on the right, and in the middle you see the fusion, so it's very similar outlines of the tumor. It seems to be sometimes a little bit bigger, and also the infiltrating parts of the tumor are sometimes more highlighted than in gadolinium. Also in the lower patient you see the same structure, really similar, but it seems to be a little bit bigger in the dnsAREX. So we also checked if this is correlating with gadolinium T1 really, and we found a significant correlation with gadolinium T1. And we also found a negative correlation with ADC values, which also speaks for this infiltration of the tumor, so that the cellularity is increased, and therefore we have higher dnsAREX signals, and at the same time lowered ADC values. We also saw this in tumor patients, so you can look in the paper, there are really 10 patients, and they all look very similar in this feature to have his resemblance with gadolinium T1. Still, it's a unique contrast because sometimes we see different contrasts or different hotspots. So here, for example, you see in the upper patient, you see that gadolinium hotspot is in this larger tumor is on the upper side, but the dnsAREX shows a hotspot at the lower part of the tumor. And also in the so we see some more substructure in the tumors, which is very interesting. So, but this is the very nice contrast. So I want to make a short conclusion. So what we did is we did all the corrections to our best knowledge to get the pure CEST effect according to the verified theory. We did B0 correction, B1 correction, T2, MT, T1 correction. So all the relaxation is compensated for. We did isolation of the different effects, and we finally did this aromatic NOE or downfield-NOE suppression. And then we see a contrast that matches very well with gadolinium contrast enhancement. So this is very interesting, and this is the major conclusion. If you do everything right, you see something that's really known to be tumor affected area, and it's a really nice outline without the need of any contrast agent. What this really is, what's the origin of that, we still don't know. It could be increased pH intracellularly, it could be infiltrating tumors, so more peptides, more cells, more proteins. So it could also be extracellular proteins because of the blood-brain barrier breakdown. So this has still to be investigated, but that it matches, that's very interesting. Further, for us it was interesting because we think now that there's a real amide-CEST contrast. Before we were like, what's going on here? There's so many contributions. There's MT, T1, everything in this MTR symmetry. But here we see, okay, there's something left over that really shows an interesting contrast. Also, we can learn and understand MTR symmetry better because we also use some upfield and some downfield signals, and maybe also in MTR symmetry there's some cancelling of this aromatic and aliphatic NOE. Also, what we can say as a conclusion is this MTR symmetry shows something, but an improved CEST contrast for these amides can be even more interesting. And maybe it's also an alternative for gadolinium contrast agents. With this, I want to thank you for your attention and all the people that were involved in the studies in Heidelberg, at the DKFZ, and at the university clinic. I quickly want to announce a resource website, cestsources.org, where we also exchange software in the evaluation to create this nice contrasts. And with that, I want to end and thank you very much for your attention.